So this might seem like a disparate combination of topics. So I'll give a bit of motivation for the kinds of theoretical and computational problems that we've been studying, which all comes from a particular biological problem. Okay, so I'll start with presenting this biological problem, and then actually going to, what I want to do with all these different kinds of machine learning topics. So what I find really exciting is, nowadays we have, through the development of all kinds of technologies in genomics, you can actually look at single cells in a very, you can, for example, look at 100,000 of single cells in one tissue at the same time. And you will see that, not only are there very different cell types, like you can imagine that a neuron is very different to a skin cell, right? But even if I take a tissue that mainly has skin cells, you'll see that there's a huge variability of cells inside this tissue. And this, in fact, nowadays we can observe cells at the single cell level by, all kinds of different technologies. For example, different kind of imaging technologies. You can stain for all kinds of stuff. But not only imaging, you can also do sequencing on the cell. So you can measure the expression of every single gene inside a single cell. Okay, so humans have 20,000 genes. So that's a 20,000 dimensional vector. And you can do this by taking, cells out of the blood and just sequence all of the cells that you have there. Okay, so that would be a data set of like 100,000 cells times 20,000 variables, and you can observe these. Okay, so imaging and sequencing, etc. And what I find particularly exciting is that not only can you observe cells, but you can perform interventions and then see what happens in these cells. So there are nowadays libraries for knocking out any one of your genes. Okay, so that really means cutting it, so setting the expression of that gene to 0. And then you can observe how do all the other genes change, or how does a cell change. Okay, so that's, I think, the first time in any area, in any application area, where you can actually perform really large-scale interventional experiments. Okay, so this is all the amazing things that comes out of biology. Now, what are the big challenges? And I will talk about one particular one. Well, getting these observations usually requires killing the cell. Okay, so if I want to do a very high content image, I usually have to fix the cell, meaning I kill it. And same for sequencing. So I cannot get both, I will call this data modalities, in the same cell. Okay, because either I sequence it, I get its expression, or I image it, but I cannot do both because both requires killing the cell. Okay, so that means that I can take a population of cells which are, say, skin cells, and I can take some out for imaging and some out for sequencing, but I will not get matched samples. Same thing is with interventions, okay? I can take a cell and I can knock out gene 1, but I then have to decide, do I want to measure it before the intervention or after? I cannot do both, okay? Because again, measuring it means killing the cell. Same for time series data. You'll never be able, right now you will not be able to get the standard time series data sets. I have a population of cells that changes over time. I can take some out at time point 1 and sequence them, but I cannot sequence the same cells again at time point 2. I'll have to take other ones and then at time point 3 other ones. Okay, so you have this big challenge of data integration, and I would say even more of translation. So what I would really like to be able to do is, I take a cell at time point 4, and I would like to ask, well, how would this cell have looked like at time point 3 were I able to sequence it or image it also then? Or same for interventions, right? I get the cell before an intervention. This particular cell, how would it have looked like after the intervention? Were I able to measure it then as well? Okay, so these are integration and translation questions. So those are, that's the big question that we've been working on now over the last years. And so first I wanted to, before coming here, I thought to main talk about causality. Now, given the talks from yesterday, I thought I will mainly talk about the second part. So the first part is about how to integrate observational and interventional data and use both in order to learn, for example, the gene regulatory network. Okay, so this, of course, if you want to learn from interventions, well, that means you have to take a causal perspective because you want to be able to predict, right? What does an intervention do if I haven't seen it yet? And the second part is about integration and translation, not between observational and interventional data, but between, for example, different data modalities or different time points, as we talked about. Okay, and so in this part, we've really come to love autoencoders. And since autoencoders are used all over the place, I'll just show you, an application or two. But then we really started asking, well, why do they work so well? When do they work? Can we actually figure out what is the implicit bias of autoencoders? How should we apply them, etc.? So here the question will really be about the implicit bias of autoencoders and can we characterize it? Okay, so on the causal perspective, I thought I'll do that short. And I'll just give you a bit of an overview and in particular tell you where the biggest open questions are, where I would love to have help from all kinds of people with different backgrounds. Okay, so how do we integrate observational and interventional data? Now, of course, causality is an old field, right? It was started about in the 1920s. But I think what is completely new is, so until very recently, everything that was done was based on observational data. Okay, so what is really new is that we have to come up with a framework, theoretical, computational, methodological, to deal with interventional data. Okay, so what is the standard framework just based on observations? Well, okay, so I want to represent my causal structure. We'll represent it by a directed acyclic graph. Acyclic because you can only cause something forward in time, right? So you cannot have any directed cycles in a causal graph. Okay, so that's how I will represent causal structures. And then, if you're familiar with Bayesian networks, that's the observational setting. Every node here is a random variable. And this directed acyclic graph encodes the factorization of the joint distribution. It encodes, the whole distribution here in the observational setting, where every node is just some function of its direct parents and some noise. Okay? And I want to keep it completely non-parametric. Okay, in particular for gene expression, that's important. So these are really any functions, so not necessarily linear, and any noise. So that's a Bayesian network, if you're familiar with it. But now what is new is that, this in a structural equation model that not only encodes the observational distribution, but also the interventional distribution. That's why I write it with these assignments here. So what a structural equation model also tells you is what happens to the distribution when you do an intervention. Okay, so we'll distinguish two different types of interventions, again, motivated by biology. So let's take the most invasive one. So I take some gene, and I just set its expression level to 0. I just cut it out, for example. Okay, so what happens? So I take this gene, I set its expression level to 0. Well, then there is no more effect of the parents, right? I'm just set to 0, so I don't care anymore what x1 does. So as you also see here in the definition, right, I'm changing the graph structure. I am now set to 0. There is no more effect on my parents, right? Before I had here x1. Now there is no more x1 in here. So those are the most invasive interventions you can do. Those are known as hard interventions. Okay. You can also randomize them, right? I can also have some noise here. And everything else I'll call a soft intervention. So if you come from a genomics background, that could, for example, be an imperfectly performed knockout experiment or any knockdown experiment, etc. Okay, so here the graph structure is still the same. All I'm doing is I'm changing how x1 acts on me, or I can change the noise. Okay, I just have to change the distribution by this intervention. Okay, and so now what are the questions? Well, causal structure discovery without any interventions. The question were, I get some samples, right, from the joint distribution. Can I learn the graph? Now I have samples not just from the observational distribution, but also from the interventions. And the question is, can I learn the graph? Okay. Now, again, in the observational setting, we know this very well, that in general, you cannot learn the full graph. You can learn an equivalence class of graphs. In particular, in the observational distribution, if you see that x is correlated with y, you don't know if x causes y or y causes x. Right? So now the question is, if you also have interventions, how much more can you identify about the graph? Okay, so I think that's the first question. It's just the identifiability question. Right? I have observational data and interventional data. Can I identify more about the graph? In particular, and this is important from an experimental design question in biology, is there a difference between hard and soft interventions? Okay, so in genomics, people have usually tried to do knockout experiments, believing that that will give you more information about the underlying gene regulatory network. So the question is that actually true? Okay. then once you know what you can identify, at least when the sample size goes to infinity, then you can ask the next question, namely, can I come up with algorithms which are consistent, right? So until I don't know what you can identify, at least when the sample size goes to infinity, you cannot come up with algorithms that are provably consistent. So only once you solve this question, then can you define algorithms that actually make sense. So we would like an algorithm where we can prove that as the sample size goes to infinity, it will actually output whatever you can learn from the graph. Okay? It will output the interventional equivalence class. And finally, and this is where I think there are all the open problems, and Henry's laughing, is optimal experimental design. So for now, we're playing with data that I just get from someone who has actually done the interventions. But the real question, and this is what I'm always asked at the Broad and from anyone who does these interventions, well, there are libraries for knocking out any of the 20,000 genes. So I can also knock out combinations of genes. So which ones should I knock out in order to learn the most about the underlying system? Okay, so this is optimal experimental design questions of interventions. Okay, so I'll just give one slide of answering these two questions and then tell you about what we know about optimal experimental design, which is really little. Okay, so just one slide. So first, identifiability is solved. So we know, and this is maybe surprising for a genomics perspective, actually, because it really changes how you do experiments. Hard and soft interventions provide the same amount of information. Okay, so it's not true that with a hard intervention, which is more invasive, you can actually gain anything in terms of identifiability. So there is a nice statistical computational trade-off. We're paying the price in terms of computation time. So it is harder to deal with soft interventions for now. We don't have any provable results like that. But in the algorithms we're using, we're paying the price in terms of computation time. So identifiability, we know exactly what you can get as the sample size goes to infinity and when you have observational and interventional data. Okay. So then the question is about algorithms. And now I'm super happy that Jean-Philippe talked before. You had exactly this kind of picture already before. Okay, so there are, as I said, right, 1920s is when causal inference from observations started. So obviously, there has been a lot of research in causal structure discovery in the observational setting. In particular, there are two classes of algorithms, and so we tried quite hard, and others tried quite hard, to get to algorithms which are still consistent by extending these, which are still consistent when you have observations and interventions. Now, there have been some algorithms described. We proved that all of them are actually not consistent. So provably, if I can very easily come up with cases where they're actually not consistent. And in particular, we were not able to extend these algorithms that have been around in the observational setting to anything that was consistent in the interventional setting. It might be possible, but we were not able to do that. So that meant that we had to come up with a new kind of or sort of algorithms that also works in the observational setting that is geared towards also being consistent in the interventional setting. And so since it's such a simple idea, I just want to tell you what the idea is, which actually led to all of these first provably consistent algorithms for learning a causal graph when you actually have observational and interventional data. Okay, so what is the intuition? It's really super simple. So I want to learn a DAG, or an equivalence class of DAGs. Okay? So what defines a DAG? Well, a DAG is defined by an undirected graph and a permutation, right? Because if I give you an undirected graph and I tell you that node 1 is smaller than node 2, well, I'll just orient all of the edges according to the permutation. Okay? Now, why is learning a DAG or a Bayesian network so much harder than learning an undirected graph? Well, it's because I have to learn the permutation. Okay? So now let me just pull apart these two, and let me just phrase causal inference as the problem of learning a permutation. Okay, so if I give you a permutation, then it's actually pretty easy, and this has been studied to get the corresponding undirected graph. So I'll just forget about that problem for now. I just want to learn the permutation. Okay, so we saw the space of permutations is a nice polytope. Okay, it's known as the permutahedron. All of the vertices are in fact permutations. Okay, causal inference is NP-hard. That's very well known, right? So what I can hope to do is something greedy. It's a huge space, as was said, but the space is much smaller than the space of graphs, where people have, the standard approaches for causal inference are greedy searches on the space of graphs. Okay, so this is a much smaller space. So what we want to do is greedy search over this space. It's a polytope, so what will I do? Well, I'll just do a simplex algorithm on this space, right? I'll start in some vertex, look at my neighbors, have some score function, and just walk there if I'm, have a score function that is at most as high as wherever I was. Okay? So that's what I want to do. I have to define some score function. Well, you can either take BIC. Now, the problem here is, of course, I need to define some probability distribution. Or, in fact, I can also do it completely non-parametrically by just counting the number of edges in the corresponding undirected graph. And we can prove that this is provably consistent and you're not getting stuck. Okay, so that's the hard part, right? If I were to enumerate all permutations, it's easy to show that the sparsest one, or if you look at BIC, the best one in terms of BIC, is actually the true permutation. But the problem is, and this is what took us so long, is that what I want to show is that I can start anywhere, and I'm just looking at my neighbors, and I'm just going to move around here, and I'm not going to get stuck anywhere. Okay, so any local optimum is actually going to be a global optimum.
Can you prove only low number of iterations or not?
No, I can. It's NP hard, right? So I will not be able to do this problem. So in particular, and how do you see this? So in particular, if the true graph is the full graph, every one of the permutations has the same score. So I'll have to go through all of them to know that I'm done. Are you trying to tell me something? Oh, okay. Yeah, so, well, yeah, okay, so I think this was self-explaining. Okay, so why can you just actually turn this algorithm very easily into something that works with interventions? Let me just explain it when you have hard interventions. Okay, so with a hard intervention, I said, if I am an intervened node, there cannot be anyone pointing into me, right? So if I'm here in some permutation, I look at my neighbors. If in this neighboring permutation, there will be an edge pointing into me and I'm an intervened node, I'm not going to walk there. So I'm actually going to cut off certain search directions given by my interventions, and we can prove that that still doesn't get stuck. Okay, so that's nice that you're actually going to gain in terms of, or reduce in terms of computation time when you have interventions. So you gain in terms of identifiability and in terms of computation time. Okay, and you can prove that this is in fact consistent also with soft interventions. And I think this is very nice, that you don't even have to tell me what your intervention targets are. I can learn them greedily at the same time as I'm learning the graph. Okay? This is super important in biology because I have all kinds of off-target effects. So I don't need to assume that I know the targets. Okay, so that's in terms of algorithms. So I think in terms of algorithms, and we also apply it, to medical kinds of applications. So I think, and just to say, why is it so important that you do something causal? Well, in a medical application, I want to be able to give you new candidates for drug targets, right? So if you just learn an undirected graph of like a difference between, in this case here, I have two patient cohorts in ovarian cancer, one with very different survival rates. I really want to get things like this, these nodes that could explain the differences between these two populations and are causal, right? Because this gives me new candidates for new drug discovery. Okay, so that's in terms of, actually causal structure discovery when you also have interventional data. Now, the last question is this experimental design question. And so this is really pretty widely open. So I'll just tell you the one paper we have and also tell you all of the limitations of this paper. Okay, so first of all, what is the problem that people care about? So as I said, so sequencing cells costs. Okay, it still costs about maybe $1 per cell. So you care about a budget. Okay, so maybe you can sequence 100,000 cells. And you also have a postdoc who has to do the experiments. So maybe this postdoc can do 4 rounds of experiments. Okay, so say now in the simplest case, I'm just going to divide up the samples. I have 25K, 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 25K in 4 rounds of experiments. So you decide in the first batch which interventions do you want to do. You can knock out any one of the genes or combinations of genes. You get to see the data. Then you decide which next interventions you want to do, et cetera, et cetera. Now you have some objective. What we looked at here is which intervention should I do in order to learn the most about the underlying causal structure. There are all kinds of other objectives that you might care about. Okay. So now you can show pretty easily that this problem is NP hard in general. Okay. So then what is nice is that you actually have submodularity. So if you already have a lot of samples from one particular intervention, of course, getting one more sample is not as important as when you have very few samples from this intervention. Okay, so now you can use submodularity to show that you can actually come up with a greedy approach for selecting these interventions, which you have guarantees on how far away you are from the optimal strategy. So now telling you what are the limitations here. So we only allow hard interventions. We don't have anything for soft interventions. We only allow single node interventions. So of course, in biology, you can do combinations of interventions. So we would need different forms of submodularity to actually get further. Also, the objective here is, as I said, is entropy, right? At the beginning, I have uniform distribution over all causal structures, and I'm trying to find the interventions that reduce the entropy as much as possible. So I do something based on mutual information. Of course, you can have all kinds of other objective functions. Then you want to define something that combines reinforcement learning, for example, with causality. Right? That would be another big area which we'll have to do if we want to get further in terms of this experimental design questions. Okay? So I think this is where we have a huge amount of open problems. Okay? So this was all about just a very brief overview on where I think we are in terms of combining observational and interventional data in order to do inference with it. Okay, and there, as I said, experimental design is, I think, one of the big areas where we really need to make some progress in. Okay, so now that we combined observational and interventional data, let's think about how to integrate and translate between, say, different data modalities. Okay, so expression data and image data, or different time points, time point 1, 2, 3, 4. And here we got to love autoencoders, and let me just show you, a particular application, and then get to the theoretical questions of autoencoders. So why are autoencoders so relevant for actually combining, integrating, and translating between different data modalities? Okay, so autoencoder, all that is important is that it is a function, a neural network that goes from RD to RD. Okay, that's what I'm going to be using, and that it minimizes some reconstruction loss. Okay, so this is what I'm trying to minimize. Okay, so say now you have many different kinds of data modalities. So you have images, you have expression data, so RNA-seq. You can have all kinds of other stuff, methylation data, et cetera, et cetera. And so these are my different data modalities. Okay, and I measure these data modalities from the same population of cells, right? Say, in this case, fibroblasts, some cell type. But as I said, I don't have paired data. Right? I don't have the image and the expression in the same cell, but I have a lot of cells, and hopefully some will be I should be able to translate between them. Right? I'd like to answer the question of, I get to see an image, how would its expression profile look like? Were I able to get it? So how can you use autoencoders? Well, so maybe you're familiar with CycleGAN, right? There's all kinds of approaches for going from images to images. Now this is different, and it is really motivated by biology. So instead of having a map going from one data modality to the other, I'm going to go to the latent space and combine everything in the latent space. Okay, because it is the same population of cells. So I have an autoencoder for each one of my data modalities, right? Going to the latent space and back, so here. And since it's the same population of cells, I'll have a discriminator in the latent space that just makes sure that I only get one population of cells here. Okay, and we have different kinds of guarantees. It was presented in ICML last year in this paper here, just on the theoretical side. If of course, now this allows you to translate, right? I can go from one data modality to the latent space and use a different autoencoder to go out to a different space. So you can do the standard things on images. Say my modalities are black-haired women, blonde-haired women, brown-haired, black-haired men, etc. I can take a black-haired woman, move her to the latent space, use the other autoencoder to produce an image of her in blonde hair, right? So all of these are generated images. These are the real images, right? But of course, I can now do this also with biological data, right? So I can actually answer questions, and we validated this. So that's in this paper here. Which is under review right now. So we really validated it by going from real images of single cells. This is the packing of the DNA to this latent space. We have analysis also of the latent space to predict its RNA-seq profile, and the other way around. Okay. So this is something that in general you cannot get in the same image. So of course you can ask, how can we validate it? Well, we can validate it for particular genes. We can just never get the full vector over here. Okay, so this actually seems to work very well for biological data. So of course, we need to understand why our autoencoders are so good, right? Similarly, I mean, we heard optimal transport before. You know, something that I have always found very frustrating in genomic or in biology is that, you know, if we think about cancer prediction or early cancer prediction, we always rely on people in medicine, right? Because they will give us a training data set that has class labels, cancer, non-cancer. And in some sense, I'll never be able to predict cancer earlier than, you know, what some medical person will give me. So what I would like to do is try to answer the question, well, I have this cell that was classified as cancerous by someone. Can I predict how this cell would have looked like at an earlier time point? So I will generate my own data sets at earlier time points and see if I already can find biomarkers, right, at earlier time points than what a pathologist can do nowadays. And so again, all we do, and I won't go, you know, in detail into this, is you can combine autoencoders with optimal transport in the latent space. And so then you can take a cancerous cell, you know, at any time point. I move it to the latent space. I have my optimal transport map that I learned, and I can ask how this particular cell would have looked like at earlier time points. Okay, and again, we validated this by actually finding new biomarkers that were not available before. Okay, so somehow, for some reason, these autoencoders work really, really well, and you know, you guys also have something with autoencoders. There are a lot of papers using autoencoders right now in single-cell biology, so the question is really we should better understand them. Okay, so hopefully that was enough motivation to study autoencoders. So now let's see what we can say about them from a theoretical perspective. Okay, so this is the problem I want to study, and I will study it in the interpolating regime. Okay, we've heard all these talks yesterday about the interpolating regime. So I will study it in the setting where I can get this loss to 0. Okay, so I have my training data here. This is the autoencoder map that is learned, and I'm looking at the reconstruction loss, and I'm in the regime where this can actually be trained to 0 loss. Okay, so now we want to know, well, what are the functions that are actually learned by an autoencoder? Okay, so let's do something very simple. Let me take the simplest setting where I have one training image and my map is linear. Okay? So then, what is this? So this problem here is the problem of solving my matrix, which is, say, A times X minus X in 2 norm. Right? So obviously, there are many solutions, right? A could be the identity, or it could be a rank 1 map, which just projects to my image, or, you know, many things in between. So the question is, what does an autoencoder do, right? All of these are interpolating solutions. So which one does the autoencoder actually choose? Okay, and probably, you know, hopefully, I mean, you would have the right intuition. So you can actually just train this. So let's train an autoencoder on one image and see what is the map that it learned. Okay, so you train it on one image and you look at what is the map that is learned. Well, you see here, whatever I plug in, the same image, the training image comes out. So that means it certainly didn't learn the identity map, right? It actually learned a rank 1 solution. Okay, so it chose by, you know, in this case, stochastic gradient descent or whatever you use to optimize, it chose to actually learn the rank 1 solution instead of, for example, the full rank solution. Okay, so the question is this a general phenomenon? So first of all, let's get some intuition, and I like it to just go to linear regression. I think you did it. So let me go to linear regression. So in linear regression, this is the nonlinear setting, but you know, it's really just the same result. So if I don't have any latent variables, I just have a linear map or a nonlinear map, then we know that if you just do gradient descent and you initialize as 0, as you did, or close to 0, I mean, in the theory, I can initialize at 0, we know that the map that is learned is, in fact, a projection. Okay, so this is well known. And so what we do here is you can actually really just generalize this to the nonlinear setting. You define something that are nonlinear eigenvectors. You define something that is a nonlinear span. And you'll see that the map that is learned, even in the nonlinear setting, is, in fact, a projection onto your nonlinear span of the training data. Okay? So now the question is, does this somehow generalize to what we actually get to see? Right? Does this really generalize to why is it that we're actually, you know, memorizing these training images? Is it just a projection onto the span of the training data? And in fact, so let's do this now for more examples than just one. So let's take 10 examples. So if you have a linear autoencoder, convolutional, fully connected, whatever you want, you'll see that actually this theorem explains everything, and we can also prove it, okay, even with latent layers, etc. So you see that, these are my 10 training images. So whatever I get out is always some linear combination of my 10 training images. So it's really just a projection onto the span of the training data. However, in the nonlinear setting, that's not what is happening. It's not a projection, a nonlinear projection onto your training data. What you see what happens is that, this is all, I just take any training images and I ordered them. I just put in, whatever comes out and whatever is the closest example, I put it into these columns. You see that whatever you put in, a training example comes out. Okay? So it's not a span of your training data, not even a nonlinear span of your training data. So, it's not that this result explains everything. And this should already have been clear here, right? Because I didn't get something in a span. I didn't get like a constant times the car. I actually got the car itself out. Okay? So the question is, why is this happening? So this is happening, I put here deep enough, okay, and wide enough, in fact, as well. So what happens if you don't go so deep? Let's look at what happens there. And this will also give all of the intuition for all of the results that come. So if you don't go so deep, and I'll explain what we have here. So here, for example, take some C4 images. So if you don't go so deep, what comes out after, this is a test image in my trained autoencoder. If I just look at what comes out, you'll see something comes out that you don't recognize as one of your training images. However, and here is where I'm using that, an autoencoder is super nice because it is a map from RD to RD. So let me just plug in the image again into my autoencoder. Okay, so I'll just iterate it. And so what you see is, okay, yes, nothing came out that looked like a training image. I'll iterate it, and sure enough, you're actually going to converge. And what this is a training image. So you'll just have to believe me. Okay. So what I can do with autoencoders, in order to figure out what are these maps that are actually learned by an autoencoder, is that I can view these autoencoders as a discrete dynamical system, right? Because I can just iterate the map. And so what we can prove, and let me parse this. I wrote it a bit hand-wiggly. OK, so you can take any autoencoder of some depth and some width. Okay. And now I'll explain what these suitable conditions are. So you take whatever your nonlinearity is that you like and the initialization that you like. Then, and this theorem is still only explaining everything for one training image. OK, that's this one training image. We can actually make it be an attractor. And how do you prove that it is an attractor? Well, we have to show that the max eigenvalue of the Jacobian is smaller than 1. Right? So this is exactly what we proved, that actually the max eigenvalue of the Jacobian at this training image is smaller than 1, meaning it is an attractor. Okay? And the suitable conditions are, we have a formula for what this max eigenvalue is. Okay? So in particular, we can show how this max eigenvalue depends on the depth and the width of your autoencoder. And that if you increase depth, given some nonlinearity that you like to take, how this will actually scale. We believe that we should be, although it's very hard, we should be able to extend this to training data that are orthonormal to each other. We still don't have that result. Okay? But that's where we're hoping to be able to generalize a result like this. So we can prove that the examples are that what the map is that an autoencoder learns when you use, this is trained using gradient descent. Sorry, I should have written this. So we only have the result for gradient descent. And this is really gradient descent stepwise, so not something like gradient flow or something like that. So what we prove is that so there are many interpolating solutions, right? I'm drawing it here for a one-dimensional autoencoder, so a map from R1 to R1. These are my training images. There are many different kinds of crazy solutions that actually interpolating solutions that go through my training images. But the ones that the overparameterized autoencoder learns is a map that is contractive at the training examples. Okay? And hence, if you iterate this map, well, what comes out is something like this. And when you have deep enough network, then we actually see this directly. Right? We saw that everything, even in one step, just immediately got mapped to the training image. Yeah.
Do you think this is happening because you have very few images to train?
It has to be in the interpolating regime, right? So, but it's well known. I mean, there are all these papers that show that even for GANs, you sometimes get to see.
Yeah, the cycle GANs.
But yes, and so even with variational autoencoders, you still get to see something about your training images, right? Where we prove it is everything in the interpolating regime. Now you can say, well, maybe this is not surprising. This is actually what we get to hear. Oh, well, it's not surprising that the training images are attractors. It's extremely surprising that in R100, any function has an attractor, right? So it's extremely surprising, right? Because I need all of the vectors in this huge dimensional space to be pointing in. Okay? So there shouldn't be attractors in these large dimensional spaces. So that an autoencoder actually learns these functions is really, I think, pretty remarkable. Okay? So this is, it could learn anything else, but what it actually learns are these contractive maps. And so now you can ask, well, what is the capacity? How do these attracting regions look like? So in fact, we can train autoencoders that have 500, all 500 training images are attractors. We prove it, right? We just compute the eigenvalue. We can do that for fully connected networks. We cannot really do it for convolutional networks. You can ask, how big are the regions around these points, right, where they're attracted to? You see here that, you know, this is a really nice form of memory, right? I put in something where I just, basically blur out a huge proportion of the space, and what you get out, if you just let it through your autoencoder, is in fact the image itself. Okay, this is up to numerical precision. I'm actually getting out my image, and I get numerical precision. I just iterate the map. So even if it is not deep enough, I'm just going to iterate the map, and I really get exactly my training image out. So there are all kinds of questions of asking, how do these regions of attraction look like? They depend on the nonlinearity you're using. Sometimes they seem to be something like Euclidean, so that's doing something like the closest neighbor in Euclidean distance. We don't know, so those are all open problems. So there is big capacity. Also, what is interesting is in these networks, we cannot prove it, but we have not found a single attractor that is not a training image. Okay, but we cannot prove it. Okay, so maybe just a word about the word memorization, since we already discussed this. For me, memorization has to have two properties. It should not just be meaning an interpolating map, which means storing the training examples. Memory in humans also means you're able to retrieve it. Okay, so this here is really also able to retrieve it because I'm just iterating the map and I'm actually going to retrieve the stored examples. So this, I think, that's why we really call this memorization, and I think only one of the two should not be called memorization, but we can have a discussion about that. Okay, so now you can start doing crazy stuff if you want. So, why going just autoencoder, so image to the same image? Well, let me do sequence encoder. I map image 1 to image 2, image 2 to image 3, image 3 to image 4, the last image to the first one. Okay, and in fact, if you do that again, and do I have 40 minutes? Yes. Okay, let me just actually show you. You can do all kinds of crazy stuff with it. Let me show you Mickey Mouse. I can memorize full movies. So you see here, I'm starting with Gaussian noise. Okay, I trained it like this, right? Image 1 to image 2, etc. And you let it play, and it immediately goes into the sequence that it memorized. And it will just play Mickey Mouse over and over again. You can memorize speech. Here is Trump. You see at the beginning you have like all kinds of noise, but now it is completely in the sequence. Right? So we memorized our abstract. You can do whatever you want, right? You put in noise and it will actually converge to whatever you trained it on. And it's actually much easier, and this is maybe something that I will, oops, that's not what you want to see. It's in fact, and this is something, interesting from, I think, a neuroscience perspective. So we can ask the following question. So what is the capacity of these autoencoders? So let me take 100 images and train it on 100 images as you usually train an autoencoder. But I can also train it differently. I can chunk up the 100 images into 10, 10, 10, 10, 10, etc., and train it as a sequence. right? Image 1 gets mapped to image 2, etc., and image 10 gets mapped to the first one. And I encode 10 sequences. I train on 10 sequences. And I can ask, well, how deep does my network and wide does my network have to be to actually get these sequences back out? Or I can do the most extreme, and I just have one sequence of length 100. And what is really interesting is that it's much easier to memorize sequences than it is to memorize single images. So you see here, right, that in order to memorize my 100 images, here we got to 99, we needed a quite deep and wide network. Of course, if you go wider and deeper, we proved that you will be able, at least for one, we proved it, that you'll be able to actually always memorize these. But here you see that, in fact, for memorizing the sequence of 100 images, you need like very, very small networks, right? Already depth 1 and some width is actually enough to memorize the whole 100 images as a sequence. And that is something that seems to be also happening for humans, right? Is that we are better at memorizing whole sequences of things than single instances. And we also have, of course, the training mechanism is still probably not very biologically meaningful, but at least the retrieval mechanism is very biologically meaningful. We're just iterating the map. Right. So, OK, so with that, let me end. So I presented our work on integrating and integrating different observational and interventional data, showed you how autoencoders have been super helpful and useful for single-cell biology. And then what we know, I think they're also a really amazing kind of framework to actually study what is the implicit bias of neural networks, because they're so nice. I can go from RD to RD, I can iterate the map, and I can actually figure out what is happening. And if you care about our biological kinds of questions of images to expression, etc., we have a lot of reviews. And with that, let me just end. And I'll just highlight that the work that I mainly presented at the end it was done by my PhD student, Adit, some of it also with Karen, and then with Misha Belkin as a collaborator. So thank you guys very much.